So, good morning, everyone, and thank you for coming today. I'm Lea Piagentini, and I will talk to you about the internship I made in the context of the Immersion project, the effects of global change on the Mediterranean corraligenous assemblages. So it was conducted at the LOV, the Laboratoire Océanographique de Villefranche-sur-Mer, and my supervisor was Steve here. Actually, I mainly focused on the temperature, so my internship could be more effects of global warming on the Mediterranean corraligenous assemblages. I will present you the context of my study and how we implemented the experimental design, what were the main results of the study, and what we learned from them. So climate change. As you may know, since the last century we are facing a fast climate change. And indeed, the IPCC is IPCC is predicting an increase of 1 degree in the best case scenario here to 3 degrees for the ocean at the end of the century. But this global warming comes also with a lengthening of the summer conditions and a multiplication of marine heat waves. So both of these events are very harmful for the marine organisms. This changes in patterns of temperature will lead to irreversible ecological transformations, such as tropicalization of marine communities, as we already saw with the invasive species, and a potential mass extinction. The Mediterranean Sea is actually a hotspot for climate change. Indeed, the future condition will be among the most distinct from the current ones in the near future, so the end of the century, because of its small volume. Actually, so the Mediterranean Sea is only 0.3% of the world ocean volume. Despite the really small scale sea, this really small scale ocean, it shelters nearly to 7% of the world marine biodiversity. And this diversity is already impacted by climate change. Actually, we saw more and more events, mass mortality events. And so all these characteristics are making the Mediterranean Sea as a priority for the research. And assessing the vulnerability of the Mediterranean marine habitats that are sheltering all these organisms is very important and even necessary. Among those marine habitats, they are the corraligenous habitats, one of the main habitats, with Polysidonia oceanica midose. So corraligenous means coral producer, and it refers mainly to the abundance of red coral that we can find on the types of bottom, but they are mainly composed of coralline algae, which are not corals, so coralline algae, which are engineering species. So they are also composed of cnidarians, sponges, bryozoan, and so on. The coralline algae are part of a highly diverse red algal order, and they are called corallinal, and they are distinguished by the presence of calcium carbonate in their cell walls. They exhibit a great structural complexity that is really important for the ecosystems because it allows microhabitats for a high diversity of organisms, and it also promotes the larval settlements of invertebrates, for example, the corals. So they are of ecological and economic importance, but they are strongly endangered by the climate change. So the aim of the study, because despite of their great importance, there is a current lack of studies that are investigating the effects of a prolonged heat waves on the corraligenous habitats. So the aim of the studies was to answer the question, will the global warming affect the metabolism of coralline algae? And to answer this question, it was first important to know that a majority of the studies that are already done on the subject are conducted on adult organisms, adult individuals. But we have to keep in mind that the response to environmental stress has to be known at each stage of life, because each stage of life of an organism responds differently, and the young individuals are more vulnerable to stress. So this is why we chose for this study to study the young individuals of Mesophyllum alternans. And why did we choose this species? It was because it was one of the most abundant in the Mediterranean, so it was really important for the rocky reefs, production, and it was supposed to reproduce all year long in the environment. So they were sampled in the Bay of Villefranche-sur-Mer at a depth of 15 meters. For what concerned the experimental design, the Mesophyllum alternans were maintained in 4 different temperature conditions. So from 22 to 28 degrees, that they are. It was temperature that it was a range of temperature that they are they are gonna experiment for longer period of time in the near future. So here we can show was organized the experiment. It was 2 water baths per temperature conditions, so 22 was our control, and for a total of 32 experimental plastic tanks. It's important for the next part. So the light, the pH, and the water motion, and of course the temperature was controlled parameters. And for each condition, we put adults in tanks. Then we waited for adults to reproduce in order to have our first generation of recruits. And when we had enough recruits, we removed the adults and we made standardized plates of recruits that we put in the new plastic tanks. So in the same way that this one. For what concerned the physiological measurement, in order to know what were the response to the organisms to the thermal stress, we conducted photo sampling in order to assess the coloration and the growth of the algae among time. So for what concerned the colors, pink was considered to be healthy algae, and whites were considered for being recently dead algae. So we also measured the surface that they occupied over the time. And for what concerned the photosynthesis and the respiration rates, it was measured by oximetry. And the recruits were placed into a little incubation chambers that we created during the study, because the organism was very little. And we measured the variation of oxygen concentration over time. So it was about 2 hours in the bright light under bright light for the photosynthesis, and 2 hours under in the dark for the respiration rates. So the rates were calculated from the slope of the oxygen concentration in the time. And so what we observed, unfortunately, that the F1 recruits from 3 water baths, so 3 water baths over 8, including a control, experienced a bleaching event during the 3rd week after we put them in the new tanks. So it was really unexpected, but for the recruits that were still alive, we could see that during the first part of the experiment, from March, during the month of March, there was a slight increase in the growth, and then after, the recruits' size seemed to stagnate and stay the same, even for the control one, 22. We were also able to see that bigger recruits seemed more able to grow. So it was before and after. Were more able to grow than the little recruits that we have at the beginning, which seemed to stay actually about the same size. So for the photosynthesis, we can see here that the photosynthesis seemed to increase with temperature, and the statistical analysis show that there were a significant difference between the control and the 28-degree treatment. And for what concerned the respiration rate, there were no significant difference, but we saw that there is a trend between the control and the other treatment of a factor of maybe about 2. So we could see that there is a visual trend anyway. So for what concern, what can we say about all these results? We saw that the young individual of our first generation saw their metabolism increase with the temperature. And metabolism is always synonymous of need of energy. And so this energy in nature is used for growth, for reproduction, for storage of energy, and for facing a stress. Here, we see that it was not used for the growth, for our little recruits, because there were no growth. And what it is important to say, it's that despite their natural ability to reproduce in the environment all year long, as we saw earlier, they were not able to reproduce in vitro. So it was not used for reproduction either. So we can suppose that it was used to face a stress and so stay alive, what is not sustainable for them. So because it was part of a bigger picture, we wanted to know if it was a good lab rat to make further research. And it is widely known that corallines are difficult to grow and cultivate for a long period of time in vitro. But we think that it's still possible, but maybe it depends on the species. Here we see that there were no growth, even in the control. We also experienced a bleaching event, and for the bleaching events, all the parameters were investigated to know why we experienced that. And we think that it was because of the light environment. Actually, there are known to be very to be very sensitive to irradiance. And actually, when the first recruits settled at the beginning of the experiment, they were in the bottom of the tanks. And then when we made standardized plates, we put them in the side of the new tanks. So we might think that it was the way that light arrived to them and maybe was passing through them. So this is something that they are not knowing in the real environment as they grow on hard substrate like rocks. So yes, the bleaching event might be due to their new light environment. And finally, there was no reproduction except in 2 tanks over 32. So it's like pretty much nothing. And as I said, the big picture was to the experiment being led on multiple generation. So to sum up, the metabolism of young coralline algae mesophyllum alternans was impacted actually by the prolonged exposure to higher temperature. We noticed an absence of growth and absence of reproduction, maybe inhibiting, maybe due to stress, and mesophyllum alternans does not seem to be the optimal species to grow in vitro. So for the perspectives for the next steps of the Immersion project, I would like to say that it was the first necessary step of a pioneering you can read it, experiment aiming to assess the responses of coralline algae to global change over multiple generations. And thanks to our foundings, we displayed that there were some technical issues that we will be able to rectify for the next steps, and we'll focus on another species, which is Lithophyllum kabioki, which has been more investigated so far, but reproduce less in natural environment. So to be continued. I would like to thank Steve Como, so my during all my internship, and so I on both of them, and I want to thank the whole team, the whole Shock team. So thank you, and thanks you for your attention.